most of the meat and potatoes of what's going to end up being, you know, in chapter 9 is section 1.
All right, guys. What do we got? We are at 5:40, so we'll get going here. By the way, I am recording here. I've got a couple students out tonight, maybe out for a little while, so I'm sorry. I just thought, you know what? We'll just go ahead and record. I got document camera's got a microphone on it there. It looks like it's picking me up just fine. So, what that means for you guys is just don't reveal any personal information, because I'd like to be able to post this on YouTube. It's a nice, simple way to share it. And if I've got student personal information, I can't post it on YouTube. That's a federal law. Okay? So, or at least I can't. Shouldn't post on YouTube anyway, but darn sure can't make it public. Right? So anyway, just letting you know, in recording, just don't share any personal information. Feel free to ask questions, right? Feel free to ask questions. I will try to remember to repeat them as they come in. You know, and with the advantage here is that this video is available and you can watch it double speed later when you're reviewing for the exam. Maybe that's helpful for you. If not, review your notes and go from there. Okay. Questions for me before we get going? How we doing tonight, guys? Everybody okay?
Well,
We're in chapter 10 tonight.
We'll get. We're not going to get done with chapter 10, but we'll get started in chapter 10 tonight. Start into chapter 10. Okay. So last time we did talk about cells needing to signal each other using ligands or molecules to be able to communicate, send signals back and forth, and then talked a little bit about what they can do with that. I mentioned that the text is going to talk a lot better about what the result of those signals are, and it talks about neuroreceptors and again, just good applications of what happens when those signals are sent and what we can do with those signals. So make sure, take some time, look through the chapter there, look back at your notes, and we can kind of go from there. Chapter 10. Focus of chapter 10 is talking about the process through which cell division occurs. Cells need to divide. Why? Why would cells, happy cells in your body, need to divide? What do you think?
Place the ones that die.
Place the ones that die. All right, we talked about apoptosis last time. Sometimes something goes wrong within the cell and it'll initiate a self-destruct sequence, basically, or sometimes cells will get hijacked. And so, yeah, or sometimes we have a cut or a scrape or some kind of disease or something like that where we've got to replenish those cells. What else? Other reasons? What's that? Could be. Yeah, just needed to replace the better cell. Yeah.
Let's say this, everything comes with a shelf life, so.
Yes. Yeah. So, yeah, absolutely. Just simply needing to replace worn out cells. If we're talking about single cell organisms, that's their primary means of reproduction a lot of times, is by simply creating clones of themselves, multiple cells through this division process. So there's a number of reasons why they would need to divide. And again, how do they know? They don't have eyes and ears. We talked extensively in chapter 9 about the receptors and the signals and being able to signal each other. And even we talked about the formation of biofilms, that microbes, single cell organisms, even they will talk amongst each other, tell each other, hey, there's only a certain amount of nutrients here. We potentially maybe get attacked by the host's immune system, and they'll work together and form a cooperative biofilm. So again, there are multiple ways they will know that they need to divide and reproduce, or different ways that they'll know, hey, there's enough of this type of cell here. We don't need any more. So that's kind of the whole purpose of chapter 9 is to make sure we're clear that cells need to be able to talk to each other and coordinate what they do. And one of the major massive things that cells do is they need to be able to reproduce. Those cells need to be able to divide and create copies of themselves. In order for these cells, whether it's a multi-cell organism or even if it's just a single-cell organism, single microbe or bacteria, they're all going to have to make essentially entire duplicates of themselves. Everything. Organelles, the entire contents of the nucleus, all of the DNA has to be copied. Each new cell needs its own copy of everything. The complete division. That's not a simple process, especially in eukaryotic cells when you look at all the organelles that are there and the amount of information that's stored and all the proteins. Quite a bit of energy consumed to go through this process. A lot of information, a lot of things need to be copied. Okay. So just quick review here. Right, we talked about, chapter 3 was it? And we talked about when we took a in-depth look at biomolecules, large macromolecules. One of those is DNA, deoxyribonucleic acid, DNA. And we didn't say a whole lot at that time about DNA. I kind of introduced the idea of the DNA is really used to create mRNA, messenger RNA, at least in most organisms. And it's the mRNA then that gets used by the ribosomes to create proteins. We talked a little bit about that process, gave you some videos, so go watch these videos. So really DNA's primary function is just storage, long-term storage of genetic material. If you played with computers much, you have any experience working with computers beyond just kind of casual user, I would suggest DNA is kind of like a hard drive. It's long-term storage, and where the RNA is kind of like the RAM. It's the temporary storage that you're actually going to work off of. If that doesn't mean anything to you, that's okay, right? No big deal. But again, DNA is a large, complex molecule made up of groups of adenine, thymine, cytosine, and guanine. Review here. You've seen this before. This is the A bonds with T, C bonds with G. Those are the 4 letters of the genetic alphabet, A, T, C, and G. RNA uses uracil instead of thymine. Okay. And we said that these, so again, the structure builds kind of this helix, which kind of looks like a spring, 2 wound springs. Where you've got ladders, or basically a twisted spun ladder. Kind of the structure that DNA will build. So again, really long, complex molecule for storing genetic information. The information we need to build the complex proteins. And we've just spent all this time in the last few chapters talking about cellular respiration, photosynthesis, and talking about all of the enzymes involved in those processes. Those enzymes have to be built. Those enzymes are proteins. How do they get built? Well, the information to build those is stored in the DNA molecule. And that DNA then makes RNA, or we make RNA from the DNA. And the RNA is what we make the proteins from. So again, if I back up just a little bit here, when the cell's going to divide, it needs a copy of everything. And one of the things it needs a copy of is this DNA spiral helix. Now, the text, I thought, did a pretty good job explaining to you, mentioning this idea, and I actually wanted to measure this. Oh, back up. It's not gonna like me, but something's board, that's okay. That's not even a meter. Your text says that if you take all of the DNA in a human cell, you line it up and you stretch it out end to end, it's about 6 foot long. I got a meter stick here, which is just a little more than 3 foot, about 3 foot, 3 inches. So I took all the DNA out of one of your cells, stretched it out. It would stretch that long. It's a lot. Why? Well, think about all the enzymes that we talked about. I didn't mention much in class. I said there's videos, go watch the videos. I'm not going to ask you to memorize those enzymes. There's a bunch, right? All those enzymes, every one of those, we need the information on how to build those enzymes. And all the other enzymes that you're going to need, all the other proteins, all the proteins that are in the, that are going to receive signals, going to receive ligands on the cell structure, on the cell surface. We talked about in chapter 9. All these proteins, we need information stored on how do we build those? Where does it come from? Right here. Yes?
I think the suggestion is only catching that board for those that are watching it.
I know. Yeah. I agree. Yeah, the comment is, you can't see all this on the camera. And yeah, I get that. I don't have 2 meters of field of view on the camera, I don't think. So but I hope the point kind of comes across. When this is on the projector, this line here is a little less than a meter, and twice that is the DNA. Now, keep in mind, it's not scale. I can't make this bar on the computer screen the same width as the DNA molecule would be. We can't see DNA molecules with your typical light microscopes we use in the lab. I'd have to go look and see if a scanning electron microscope can even see DNA. I'm not sure that it can in a single strand. I don't think it can, actually. So it's very small. Now, just to put this in perspective, as best I could get on this scale, this dot that I've got right there on the screen, yes, this one, that is 1,000 times larger than the typical human cell. So if the scale here is that the DNA is twice as long as the box I've got on the screen, because that's all the room I've got, and that dot that I'm pointing to there is 1,000 times bigger than your typical cell, well, how the heck does that happen, right? That's crazy. Well, that's what we do. That is actually what we do. All right, let me see if, so how do we fit all of this DNA in something this incredibly small? We have to. Well, life has found a way. Organization. Don't copy this down yet. We'll come back to it. But talk about the terms here. If I do have this graphic, we'll show again. We'll talk more detail here. But again, you can see the DNA molecule here, just your standard DNA, and it gets wrapped around this protein, and then that gets bound up, and then further and further organized. And they're showing you here with these little boxes. They're saying, hey, look at this little strand. It's right here. And then you can take this little complex here, and it's about this size here. Anyway, that's okay. I think that's helpful. If you haven't already watched it, this video and the text, let's go watch this guy real quick. I'll check the speakers, make sure they're going to work for us. Hopefully we don't get feedback on the microphone. Oh, that's going to be interesting.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome.
I've got all these in the
Multiple
slides.
nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
All right, so guys, this video right here, I would suggest watch that one a few times, especially when you're reviewing for the exam. That one is in your text. Find it, right? It's within a minute and a half. They get a lot of information packed in there. That's the reason I wanted to go ahead and show it here. A lot of information. Some terms for you. Just gather some up. They talked about most of these in the video. The histone is the protein that the single strand of DNA is going to curl or coil around. When the DNA is coiled around the histone protein, we call that coil a nucleosome. Chromatin, I mentioned this in the video, that is, you got a fiber of these nucleosomes. The DNA is wrapped around histones, forming nucleosomes. You get enough of those together, bound up and wound up, that forms the chromatin. And enough of those get together and they will actually wrap around and tighten up, forming chromosome. There's additional long proteins involved that the chromatin wraps around. And how do we get this down into a single cell, into the nucleus of a single cell, not just inside the cell, inside the nucleus of the cell? How do we get all of that information in one place? Pack it, and pack it, and wrap it, and pack it. Think about if you took a rope, twine, if you're ever taken rope and untwisted it, there's multiple fibers, there's big fibers within that, right? And then a lot of times so if you take a piece of rope, there might be 5 or 6 smaller lines in that one piece of rope. And then within each of those 5 or 6 smaller lines, there's a whole bunch of strands wound up. Similar process. Not the exact same, but similar process. Hopefully it's kind of a nice analogy most of us can relate to. Okay. So again, just kind of visually, this graphic from your book. DNA just by itself, and then we wrap the DNA around a histone that generates a nucleosome. Chromatin is where we've got a group of these nucleosomes stranded together. And we keep binding those and winding and wrapping those up, and eventually we get the chromosome. All right. Now, a lot of times you've probably heard this term chromosome before. It's a lot of times we'll present it kind of as this unit. I put unit there in quotations, kind of this unit of DNA. And yeah, that's fair. When we actually see DNA, we see it in a chromosome form, and they mentioned that in the video, that DNA, when the cell's just going about its normal day-to-day operations, normal business, the DNA is probably not going to be in this chromosome packed up real tight form. It's just going to be in the nucleus, being utilized, being read, turned in, RNA copies being made from it. And that's a little hard for that to happen when it's all tightly bound up in this chromosome form. But chromosome is normally how the DNA is organized within the cell. Especially when the cell's ready to divide, that's when these chromosomes actually become visible. We can actually see them. The cell goes through the division process. All right. Now, some chromosomes pair up. They go together. There's pairs of chromosomes, or sets. We call this the homologous chromosomes, or pairs of chromosomes. And basically, for these pairs, for organisms that are reproduced sexually, where you've got a male and a female, or two parents, each pair of chromosomes, one set comes from one parent, one set comes from the other parent. That's the reason we have pairs of chromosomes. Each chromosome contains this similar information. So the genes for your blood type, genetic information for your blood type, A, O, and B, that is contained on one particular chromosome. You have to go look that up where that's located in humans. That's easy enough to find. But the information for your blood type would be in the same chromosome, in the same location within that chromosome from both your father and from your mother. And the combination determines your blood type. If I get an A to the best of my knowledge, I'm O. I've never actually been tested in a lab. I think when I took microbiology in college, they had us do a blood test analysis. I'm almost certain I was O at that time, that's true, and I'm always O, it's not at that time. Which means I had to get an O from my father, which my dad is O, found that out this weekend. But I had to get an O from my mother, which she remembered her dad was O, so it makes sense. So it's most likely I am O. But again, that means the chromosome from my dad and the chromosome from my mom, both at the same location, contain genetic information for O blood type. If my mom, who is A, if she had given me, I think my sister is A, if she had given me her A chromosome instead, I would have been A type. Okay. So pairs of homologous chromosomes, same length, contain the same genetic information. You just get one from each parent, and that's how we get diversity. So each cell in a species has a certain number of chromosomes. Normal cells within humans have 46 chromosomes. And again, you've got homologous pairs, 23. And we're going to talk a lot more about genetics, heritability, and things like that as we move forward. Not necessarily our focus here, but it's really important. It's good to kind of lay some foundation. Now, one of those pairs, of course, is the sex chromosome. That's the one where one chromosome, for males, one chromosome is going to be different from the other. Again, more on that later. Okay, 23 pairs of chromosomes for humans. Humans are grouped. We are primates. Humans are primates. Two groups of primates. You've got your great apes and you got your lesser apes. No, it's not true. Two groups of apes. You've got your great apes and your lesser apes. And you've got primates, and so we're separate from. You've got primates, and you've got monkeys, which are old world and new world monkeys. And you got lemurs. I forget if I'm leaving anything out. I'm sure I'm leaving something out. Somebody on the internet is going to blast me. That's okay. Anyway, humans are great, or primates. Very similar to the other great apes, that's chimpanzees, gorillas, orangutans. They all have 48 chromosomes, 24 pairs. Now, the number of pairs and the number of chromosomes don't necessarily dictate the complexity of the animal. The domestic dog, as best I could find, has 39 pairs or 78 chromosomes. Sunflower, I want to say a red fox, which I thought was weird that's a canid species, and it only has like 34 chromosomes. I didn't trust that, so I didn't put that on there. I didn't verify that, but I thought it was odd that I found a few sources that suggested a sunflower and a red fox have the same number of chromosomes. I did just happen to see what organism has the most chromosomes, and Found a couple sources that suggest that in the Guinness record for most chromosomes is the adder's tongue burn. 1,400 chromosomes. Strange. Anyway, the point there is each organism can organize their DNA into chromosomes. How many chromosomes they have? Well, it depends on the species, depends on the type of organisms. Humans have 23 pairs of chromosomes, 46. We can see those when a cell divides, and this is what we call a karyotype. This is just an image that we can take of right at the point when the cell is being divided, those chromosomes are visible. What we can do is we can take, and this is from your text, and they describe how it's done. They place some fluorescent dye on the different chromosomes. They're able to dye the chromosomes with different fluorescent dyes to show color contrast. They don't come in these cool colors like this. You have to actually generate that image. And then what you can do is once you've taken a picture of the cell while the chromosomes are exposed during division, then we can cut that picture up either digitally or have it on cellophane and cut it up and arrange those in this fashion. Okay. All right. Now, again, a regular cell is going to have pairs of chromosomes. But we have special cells we call gametes. These are cells used in sexual reproduction. We talked last time, we talked about single cell organisms signal each other to communicate with one another. Right? We talked about cellular communication. We talked about single cells communicating. We said that interestingly, some types of yeast, which are single cells, will actually go through a sexual reproduction process where they will actually generate haploid cells. A haploid cell is gamete or a sexual reproduction cell where instead of a pair of chromosomes, it has half the chromosomes. It has a single set of chromosomes. So in the case of humans and most other animals, there's going to be a sperm cell and there's going to be an egg cell. So each sperm cell is going to have, for humans, 23 chromosomes. And each egg cell in the female is going to have 23 chromosomes in those. And so when the sperm and the egg cell combine, each one brings a specific set of chromosomes. They pair up, and that genetic information is used then to create reproduced organisms. So once you put two haploid cells together, you generate a diploid cell that contains a complete set, a complete number of pairs of chromosomes, one from each parent. All right. So, why is this all important? A lot of information about DNA there. Well, because DNA is one of the most important things that has to be duplicated or copied for cellular division to occur. And again, the focus of chapter 10 is to talk about process through which cells will live and go through in order to divide and create more cells. It's really the focus of chapter 10. So we have to spend a lot of time, or a good amount of time. Let's make sure we understand how important and how complicated copying all of the DNA is and how that's organized. We do also, again, as we mentioned, you have to have a copy of everything. So all the organelles must be copied as well. All the structures. It's a long process. You don't have to write all this out. We're going to talk about the first block here today. It's a long process, and what we normally do is we divide it up into 3 groups, 3 blocks. So your interface the gap 1, the S phase, and gap to G2. Mitosis then, I'm not going to get to mitosis today. Mitosis has multiple phases. Probably we'll talk about these. Prophase, metaphase, metaphase, anaphase, and telophase. And then cytokinesis. ESFs. Cytokinesis. We got graphically. Diagram from your text, I thought you did a fantastic job explaining kind of this is a timeline, or basically the amount of time roughly that many cells will go through this process. It's not evenly distributed. See, we're saying that most of the time is spent in this interphase, working our way up to the phase through which mitosis is, where everything is divided. We'll talk about this in a second, actually. I guess I shouldn't draw it here. It's on the slides. So again, all this time right here, G1, S1, G2, all of this time working our way up to the point where we divide things up and then actually cut the cell in half. That process actually goes pretty quick. It's getting ourselves organized, getting the DNA all coiled up and bound up and put into chromosomes so it's easy to split as well as replicate. That takes time. And that's what these first three phases are all. We group into interphase. So here we are. That's what I was saying. I'm not going to draw that on the board. Let's just talk about this. Talk about this on the next slide. So interphase is where the cell goes through some growth, gets big enough so when it cuts in half, it's appropriate size. Everything grows a little bit, but its main goal is to try to store up enough energy and enough building blocks to actually make copies of itself, copies of everything. Mitosis phase is where, really kind of the focus during mitosis is where we take the contents of the nucleus, long DNA, we rearrange it, we organize it, and rearrange it so it's easy to then split it in half. Because each cell needs to get half of the information, or each cell needs to get an exact copy of the information stored in the nucleus. If something gets mixed up along this long chain of DNA, any of the chromosomes, if anything gets mixed up, the resulting cell now has a mutation, may or may not be viable, may or may not survive. So it's a very important process that each cell gets an exact copy of the DNA information. That's mitosis. Complicated process. And this cytokinesis is where the cell, once all of the components of the cell are on opposite ends of the cell, so one set of DNA is on one side of the cell, one set of copy of the DNA is on the other side of the cell. The organelles move, copies of the organelles move to one side. You have enough information, enough capacity for two different cells on different sides of a single cell. cytokinesis is where the membrane gets cleaved. In animal cells, it basically just kind of it talks about this protein ring basically comes in and kind of squeezes it until they cleave off. In plant cells, it actually kind of has to build a bridge in the middle of the cell and then build a cell wall on the bridge. And then once the cell wall's in place, now I've got two cells. The cell walls, for plant cells, because of the cell wall, it's a little more, the cytokinesis is a little more complicated. So we talk about this interphase just a little bit. Let's talk about these different phases. Not too much to say, and there's not a whole lot of text, but we can talk about what they do and why they're important. In the G1 phase, what we call the first gap 1 phase, the cell is trying to gather enough energy and enough of the components that it's going to need to build copies of all the DNA, copies of all of the organelles, and within DNA and within all the organelles, there's all these proteins that are going to be copied. Need a lot of energy to do this copying process. So G1, it's just gathering up what it needs to make 2 of everything. The S phase is where the chromosomes themselves are actually duplicated. The duplicate of chromosomes, called a chromatid. That's where you've got 2 chromosomes. So chromatid is where you've got 2 chromosomes together. We'll come back here. There's a nice image here. This is essentially a chromatid structure. It's actually, this chromosome has been copied or duplicated. One side is a chromosome, the other side is a copy of that same chromosome. This is what we call a chromatid structure. It's 2 chromosomes, exact copies of each other. And what happens is when we go through mitosis, this gets cleaved, this gets the connection between the 2 copies gets dissolved. One half goes to one side of the cell, the other half goes to the other side of the cell. Okay. So in the S phase, normally we have 1 chromosome. We go through and make an entire copy of each chromosome and bind them together in what we call a chromatid. And those chromatids then can be, which is what you see in this image here. No, those appear to be chromosomes. I wouldn't call those chromatids. Okay, sorry, just kidding. You can see sometimes karyotypes where they are showing you the chromatids before they get separated. Those are not. Chromosomes during the S phase get duplicated. Second gap, the G2 phase. So after we spend all this energy making copies of these chromosomes, which is not easy for the cell, it takes a lot of energy. And we've got to save up more energy, put more energy in the cell and begin the process for mitosis, because it's moving all those chromosomes and making sure each new half is going to have 1 of every organelle. It's going to take some energy for the cell to move all that stuff to each end of the cell so that we can cut it in half once it's done through the last process, cytokinesis. Now, there is kind of this special phase, the G0 phase, the 0 gap phase. That's when the cell's just kind of sitting idle, if you will. It's not actively getting prepared for division. Not actively preparing to go through mitosis. Some cells are perpetually stuck in a G0 phase. Your text gives a couple of good examples. In a mature organism, often the heart cells, the cardiac muscle cells, are not normally reproducing. They're normally, unless one just happens, unless a substantial amount gets worn out, they normally don't. It's not necessary that they divide. So once the heart has grown to its final size, they'll tend to stay in a G0 phase. Most nerve cells do not reproduce.
That explains why when you get nerve damage, there's really no going back from it.
Yeah. So we figure out how to turn on, send the signal to those cells they need to divide or reproduce, or figure out how to put new ones on. I mean, or replace it with electrical sensors. Right now, those are kind of the three main options. Can we replace it with some kind of external tool? Yeah, when you have nerve damage, science is working on solutions, but unless there's and you can also, sometimes physical therapy will allow your body to retrain how to use whatever nerves are still there, or use the signals, whatever damage signals are currently coming from those nerves, how to reinterpret those. So yeah, no, but very much so, yes. Typically nerve cells are stuck in a G0 phase. There's no signal that tells them they need to start preparing for division. Until we figure out how to change that and not create cancer when we do. If a cell continues to reproduce uncontrollably, and if there's other damage within it, that's one way cancer gets caused. So being stuck in G0 phase is not a bad thing when you don't need more copies of it. Okay. All right, guys. I hate killing you with PowerPoint. I don't like it, but it is, sometimes that's what we got to do. Okay. This unfortunately is one of those classes where it's like, as I looked at this, it's like, you know what? Not going to go the whole time. I mean, we could talk about mitosis and the phases today. I'm seeing some head shit. No. Let's break this chapter in half. We're doing okay on time. We're kind of at a nice point in the semester. Let me stop recording here and let that. All right. Thank you guys for joining.